Hi. Well, good afternoon, everyone. Welcome you all to the EMGS Bioinformatics Challenge 2021. My name is Max Leung. I'm the host of this final round competition, and I would like to welcome you all to participate in this afternoon's event. So this afternoon, we are very excited to have speakers from 4 finalist teams in the EMGS Bioinformatics Challenge. We're very excited to have them to talk about their research this afternoon. Now, before we start, let's first go over the format of this final round competition. Now, each speaker will present the team's work for 15 minutes, and this will be followed by the Q&A section for 8 minutes. Now, in the Q&A section, both audience and judges can ask questions. And for the audience, if you guys would like to ask any question, please do put in the question in the Q&A box. Both myself and the judges would keep an eye on the Q&A box and ask the question to the speaker. Now, each of the teams will be judged on the following 4 criteria. This includes the model performance, as well as the identification of a novel genotoxic modes of action, the effectiveness of data visualization, the originality and innovation of the research, and finally, the speaker's performance in response to the question during the Q&A section. Now, I also want to express special thanks to our judging panel who are participating in today's competition. And the result of this competition will be announced at the EMGS News after the virtual EMGS meeting. Now, with that, I would like to introduce our first speaker, Joseph Bundy from the U.S. Environmental Protection Agency, and he will be talking to us about his work on the predicting molecular initiating event from high-throughput transcriptomic screening using machine learning. So, Joe, can please do pass over the share screen. Thank you so much.
Can everyone see this? Can you see this, Max?
Yeah, it is perfect. Yeah, thank you so much.
Okay. It's full screen, right?
Yep.
Okay, thank you.
Thank you.
Thanks for that introduction, Max, and thanks for inviting me to speak today. The title of my talk is Predicting Molecular Initiating Events from High-Throughput Transcriptomic Screening Using Machine Learning. So this is just the standard disclaimer saying the views expressed in this presentation are mine and don't necessarily represent the views of the U.S. Environmental Protection Agency. So a current challenge in chemical hazard identification is that we don't yet have a complete understanding of the possible ways by which chemicals can impact biological systems. And so as an example of this is about 883,000 chemicals registered in the ComTox chemicals dashboard, but many of these chemicals have limited associated chemical safety information. So a solution to this challenge is that we can develop new approach methodologies such as high-throughput transcriptomics and combine these approaches with machine learning to help identify molecular initiating events induced by chemical treatment for hundreds or thousands of chemicals in a single experiment. So what are molecular initiating events? MIEs are a concept presented in the adverse outcome pathway paradigm, and an MIE is the first event by which a chemical impinges on a biological system, causing a molecular change, which can then result in downstream key events, ultimately culminating in an adverse outcome. So our approach to predicting MIEs was to use gene expression data. And to that end, we used the Lynx L1000 gene expression data set spanning about 20,000 chemicals. And we integrated those data with RefChemDB, which is a database of chemical target interactions. After integrating these data in a machine learning framework, we trained binary classifiers to predict activation of distinct MIEs in response to chemical treatment. We trained individual classifiers to predict activation of a single MIE. And so by training a family of classifiers, we can predict activation across a spectrum of different molecular initiating events. This is our data processing and classifier training workflow. Again, we start by integrating data from the Lynx L1000 CMAP dataset with RefChemDB. And from this integrated data set, we exclude a subset of data as exemplar chemicals and profiles. So these are chemicals that are associated with distinct MIEs that we want to model, but that we excluded from classifier training for the purposes of downstream validation. We then trained individual classifiers on specialized MIE data sets. We used an 80-20 split to separate training data from a small holdout data set. And then we trained classifiers using 5-fold cross-validation in the CARET package in our statistical framework. We use then the holdout data to generate an accuracy assessment and also simultaneously performed training of 500 null classifiers for the purposes of empirical significance testing. So we integrated this empirical significance testing approach in our accuracy assessment to identify a subset of classifiers that are candidate high performance classifiers. We then cross-reference those with predictions from our training excluded exemplar chemicals to identify a final set of confirmed high performance classifiers. This is a schematic of a MIE training data set, and I'm going to use estrogen receptor inhibition or ESR1-2 inhibition as an example. So the way we generate these data sets is we first generate a collection of gene expression profiles from links associated with chemical treatments that are linked to our MIE of interest in RefChemDB. And so you'll notice that there's multiple gene expression profiles for each one of these chemicals, as there are multiple gene expression profiles and links for some chemical treatments. So after generating this MIE active set, we generate a size matched MIE inactive set. And we do this by randomly selecting chemicals that are associated, or randomly selecting profiles that are associated with chemicals that are not associated with our MIE of interest. And so then after generating both of these sets, we can train a classifier to discriminate between the two sets. And then afterwards, we can predict using an unlabeled gene expression profile, the likelihood that that profile is associated with the active set or inactive set. And then we can extrapolate the likelihood that that chemical treatment activates the MIE or not. There's a variety of cell types present in the Lynx L1000 data with a variable number of gene expression profiles. And so the cell type with the most, or the cell line with the most gene expression profiles is the breast cancer derived MCF7 cell line. And so we focused on this cell line to develop this methodology. So in MCF7 data, there were 51 MIEs with sufficient data for training based on 2 criteria. Valid MIEs must be linked to at least 5 chemicals in RefChemDB, and these MIEs must also be linked to at least 50 gene expression profiles and links. We then train classifiers using 3 different feature types and 6 different classification algorithms, half of which were support vector machines. So the first question we asked after training these classifiers was, how does internal accuracy for these classifiers vary as a function of training feature type? So these are the 3 types of training features we use. We use all genes, which is the 978 landmark genes and links, plus the abundance of an additional 11,000 genes whose expression was inferred from these 978. We also train classifiers using pathway scores for about 900 pathways. This was calculated with in SIGDB and SSGSEA. And then finally, we train classifiers on a third set of features based just on the landmark genes, just these 978 genes measured in the L1000 assay. And so this figure is kind of busy, but what I mainly want to point out is that it's broken down by classification algorithm. This is k-nearest neighbor, multi-layer perceptron, SVM linear, and it's also broken down by training feature type. So landmark genes are shown in blue, pathway scores are red, and all genes are black. And each one of these distributions represents internal accuracy from 51 MIE classifiers. And so across our training algorithms, we see that the blue distribution or landmark genes outcompete the red and black distributions significantly. So we went with landmark gene-based classifiers moving forward. So after identifying landmark genes as a good feature type, we then explored how internal accuracy varied as a function of classification algorithm. And so we compared our internal accuracies across algorithms and found that our SVM model with a polynomial kernel seemed to perform the best. But then we did a subsequent analysis where we compared holdout accuracies to internal accuracies across these different algorithms. And the reason we did this is we wanted to make sure that none of these algorithms generated holdout accuracies that were significantly lower than their internal accuracies, as this would be evidence of overfitting. Because if the classifier doesn't perform as well on data it hasn't seen before as it does on data on which it was trained, it would be overfit. And so there's only one algorithm that showed evidence of this, and that was SVM poly, which again was our looked like our top performing algorithm. But because of this, we actually excluded it from further analysis. So we further validated our landmark gene-based classifiers after removing SVM poly based classifiers with empirical significance testing. And the way we did this, we took our original data set, or again, using estrogen receptor inhibition as an example, and we trained a classifier using this true data set of chemicals associated with ESR1 inhibition and not. But simultaneously, we generated 500 null classifiers based on this template. And the way we did this is we took these chemicals that were associated with ESR1 inhibition, and we replaced them with chemicals that were randomly selected from links. And we did this on the criteria that these replacement chemicals must have at least as many gene expression profiles as the original chemical that they replaced. And so what this allowed us to do was train a classifier based on the same amount of information, but basically trained on noise. And we did this 500 times, and this allowed us to compare the internal accuracy from our true model with 500 null accuracies, which build up this distribution with which we could then use to put context on the internal accuracy shown here in the dotted line with this distribution of null accuracies. And so we calculated empirical p-value by the proportion of null accuracies that outcompeted our true accuracy. And in this case, none of them did, so the empirical p-value was 0. This is a table showing the performance statistics for our 20 candidate high performance classifiers with the MIUs shown here on the left. And all of these classifiers passed empirical significance testing with a p-value of less than 0.05, meaning the true classifier outcompeted at least 95% of its null counterparts. And we further validated these classifiers by generating predictions on exemplar chemicals. These are exemplar percent ranks for exemplar chemicals that are matched to their respective MIE. And so ranks of 1 or ranks about 1 reflect that the exemplar chemical was the top scoring chemical for that respective MIE. And so we identified 9 classifiers that met these criteria with an exemplar chemical rank of at least 9 or greater. This is a heat map of those 9 confirmed high performance classifiers shown in columns with all the exemplar chemicals in the study as rows. And so the degree of shading for these cells indicates the percentile rank. So dark cells indicate a percentile rank that's close to 1. And asterisks indicates an intersection between an exemplar chemical and the high performance classifier for which it was annotated. And so what I mainly want to point out is that asterisks fall in this heat map on cells that are dark, indicating that high performance classifiers successfully predicted their associated exemplar chemicals. But there's also some exemplar chemicals that generated high ranking predictions for MIEs for which they're not associated. So for example, methotrexate generated a high ranking prediction for top 2A inhibition, mTOR inhibition, and ESR1 inhibition. So one explanation for this is that it's likely that some of these exemplar chemicals, the mechanism of action of some of these exemplar chemicals likely converge with the downstream molecular pathways of some of these MIEs. So methotrexate, for example, is a chemotherapeutic agent and is known to halt cell proliferate or slow cell proliferation in MCF7 cells. On the other hand, while it doesn't interact directly with these MIEs, activation of these MIEs is known to be anti-proliferative in MCF7 cells. So in conclusion, we trained predictive models for 51 MIEs by integrating gene expression data with RefChemDB chemical target labels, and we explored factors that affected model accuracy like feature type and classification algorithm. We identified nine MIEs that were modeled with high performance classifiers. So in the future, we want to compare classifier performance between classifiers that are trained on data generated from different cell lines. This is actually work that's ongoing, but not yet finished. And then we'd also like to work on predicting MIEs using other HTTR data sets in addition to the Lynx L1000 data. So I'd like to thank the contributing authors, both within and outside of the agency, as well as the agency itself, and especially the computational toxicology and bioinformatics branch. And I'm happy to take any questions.
Thank you so much, Joe. So this is now the Q&A section. So for those of you who are in the audience, please do submit your question to the Q&A box. I believe we already have one question in the Q&A box. So let me go ahead and ask. So this question is about the classifier. Do the classifier not account for exposure duration or concentration?
That's a great question. So the data in Lynx the data available in Lynx are derived from a variety of different, actually, let me go here. The data in Lynx are associated with a variety of different exposure durations and concentrations. And so it's challenging because these, the gene expression profiles aren't available in necessarily a systematic fashion. So not every chemical is surveyed in Lynx or tested in Lynx with the same exact exposure or concentration criteria. So we made the conscious decision to basically treat them all the same because we didn't have really a systematic way of going through and selecting, specific concentrations or specific time criteria. There are other investigations of Lynx that have gone so far as to collapse all the profiles for a single chemical. There's also been investigations to, for example, limit it to a single concentration of 10 micromolar, if I remember correctly, which is a predominant concentration. But yeah, we basically took all the gene expression profiles in Lynx regardless of duration or concentration.
Thank you so much, Joe. So I believe we have another question, but Kevin, you have a question? Yeah, go ahead.
Super, super. I really enjoyed your presentation. That's very cool. So in the crosstalk that you discovered, can we take that as something very positive, that we can learn new mechanisms of action? And can it also tell us something about, if we were testing particular agents sort of ahead of time, discover crosstalk we hadn't anticipated?
Absolutely. So the long-term goal of this project is to, at least as I see it, is to prioritize chemicals for which we know relatively little for further screening. And so in that aspect, identifying a likely MIE for a chemical that not as a lot known about is the long-term objective. I do think it is likely that we're probably going to find some crosstalk. Sorry, let me advance through all these between chemicals that we do know a lot about for some of these MIEs. In some regards, this is a benefit and a limitation in that, we're trying to generate classifiers for specific MIEs. On the other hand, findings like this, I don't think it's likely that methotrexate impinges on any one of these distinct molecular pathways, but shares a downstream effect on it. So it's kind of cool, I think, the idea that we can resolve some previously unknown actions from some of these chemicals, but it's going to be difficult to resolve it from this molecular crosstalk downstream, which may or may not be informative, I guess, depending on our objectives.
So as a follow-up, could the crosstalk patterns be an additional piece of information for classification?
Absolutely. I definitely think so, because to a certain extent, yeah, we could prioritize other MIEs or other possible mechanisms of action or key events or adverse outcomes that should maybe be looked at for a specific chemical, even if we don't necessarily think that that chemical activates the MIE. That's still information that I think could be useful for downstream. And so combining information from different MIEs from different MIE predictions might be another way to help guide our prioritization of chemicals for further screening.
Okay, thanks, Joe. So we have one question from the audience, and then I think Tom and three questions, and then I think Tom, Amy, and Laura can go. So one question from the audience is, have you tried the ensemble techniques?
So we have, I interpret that to mean, have we tried ensemble prediction techniques? Which to me means integrating predictions from multiple classifiers to generate a prediction that's more robust. And so we actually just started giving this a try. So as I said before, we generated predictions on 6 different classification algorithms. And so right now we're saying, okay, one of these algorithms generates the highest accuracy for each MIE, even if multiple algorithms actually pass empirical significance testing. And so in the data I showed you today, we actually just take the classifier that performed the best based on internal accuracy and use that. What I've started doing is going in and looking at taking the predictions from all the classifiers that passed our criteria, and then integrating their predictions together in hopes of making a more robust prediction. Disappointingly, so far, that hasn't actually turned out to be the case. The integrating predictions from multiple high-performance classifiers doesn't actually seem to make these predictions more accurate. But I'm still working on that, so I'm not convinced that I've given that its due attention.
All right, thanks. We have 2.5 minutes left. So Tom, do you have a question? Yeah.
All right, I'll go. There's some of the questions in the Q&A are more informative than mine, probably.
Okay, you can go with that too. Yeah.
Since I'm a judge, I'll go ahead and ask. So one question is related to the landmark genes, and I wasn't completely clear. Are they classifier specific, or is it the same landmark genes being used across all MIEs?
That's a great question. So these are not. They are the same genes used across every MIE. So the links data are in the form of these 978 genes that are measured on the assay. And then, there's a lot of additional information, these inferred genes, which we can also train classifiers on, but it's kind of with a caveat that these genes, their expression wasn't directly measured. It's extrapolated, and this is done on links, not by me, or by the links CMAP consortium, not by me. And so we generated these classifiers based on those 978 landmark genes, and it was the same genes for every classifier. So I think where you're going with this is, have we looked at using specific genes for predicting an MIE based on previous knowledge that, okay, this gene set is associated with this MIE. It's probably going to be more informative than just taking all genes. And so that's something we're looking at doing that I haven't done yet. I've just done the 978 landmark genes so far.
Yeah, I also kind of wondered in reverse if that could become a self-fulfilling prophecy. You
Right.
find what you look for. But that actually leads to the question that's in the Q&A. I'll just go ahead and ask it, which is related to the inferred genes, because I understand it better now after your answer. It says 92% was inferred, only 900 were measured. Do you know how well the inference compares to the actual expression of those genes?
I think it compares relatively well. There's a subset of inferred genes that links flags is not well inferred. I don't think I filtered those out. I think I took all the inferred genes, but that information is available in links. So they did this process where they measured the full genome by some method, and then they compared it. Those actually measured values with their inference. I think they got a pretty good assessment. I want to say it was around 90, but I don't really want to misspeak, so I can't remember. But I want to say that the estimations were pretty good.
Okay. Well, thank you very much, Joe. So with that, that would be the end of the Q&A section. And I guess, Joe, I would just let you to respond to Amy about the availability of the MIE result on the chat box. I mean, if you would be willing to share it. Let's move on to our next speaker. All right. And thank you, Joe. Thanks a lot for the presentation.
Thank you.
Thank you. Yeah. Our next speaker is Adrian Green from North Carolina State University. He will be talking to us about his work leveraging zebrafish high throughput screening data and conditional generative adversarial network to advance predictive toxicology. Thank you so much, Adrian.
Thank you, Max, and thank you for the opportunity from the organizing committee. So my name is Adrian Green. I'm currently a postdoc in the lab of David Reif at North Carolina State University. And it's my pleasure to talk to you today as part of the 2021 EMGS Bioinformatics Challenge about leveraging high throughput screening data, deep neural networks, and conditional adversarial networks to advance predictive toxicology. So currently there are 85,000 chemicals registered with the US Environmental Protection Agency that are either manufactured in or imported into the US. Of these, approximately 44,000 have rigorous toxicology data, leaving over 80,000 chemicals untested. So to address this huge data gap, high throughput screening assays and computational methods are vital. So there are a number of options available for screening these environmental chemicals. Each of these offer a range of pros and cons in terms of throughput, cost, human relevance, specificity, and complexity. So biochemical or cell-based assays offer higher throughput, lower cost, and more specificity, but sacrifice complexity and human relevance. In addition, toxicological endpoints related to behavior or development are difficult to measure in these purely in vitro systems. On the other hand, higher order mammalian models offer increased complexity and relevance, but are more expensive and lower throughput, which is why the zebrafish have emerged as an extremely useful high throughput screening model, as they complement both the targeted approaches and provide systemic data. So to maximize the complexity and human relevance of this model system, zebrafish husbandry practices are followed to maintain a genetically diverse population. This ensures that both the sensitive and resistant fish are present in the population. It reduces the idiosyncratic responses and improves the translatability of these assays to humans and further to ecological health. So as shown in the diagram here on the right, in a diverse population, a chemical exposure can result in a range of responses, including no effect, moderate effect, or a severe effect. In this case, examples of like kidney damage, or further on, actually looking at cancer and some other severe effects. So therefore, maintaining a genetically diverse population, we're able to better capture these responses and actually make inferences. So in addition to the high throughput screening efforts, computational approaches to bridge the toxicity gap have been developed utilizing chemical structure, with quantitative structure-activity relationships and read-across being the most common. The recent advances in machine learning, such as deep neural networks and conditional generative adversarial networks, offer us an exciting opportunity to bridge this gap that have not been thoroughly explored. So the aim of the study is to more thoroughly explore these and to answer the question of, can we actually infer toxicological data or predict toxicity based on chemical structure? So as part of an ongoing effort, Robin Tanguay's group at Oregon State University has developed a high throughput screening system to assess the neurodevelopmental effects of environmental chemicals. As part of the study, they took 32 zebrafish embryos and exposed them to one of 1,000 chemicals at one of 6 concentrations from 6 hours post-fertilization to 5 days post-fertilization in a 96-well format. These zebrafish were then assessed for mortality and 17 morphological defects and abnormalities at 5 days. And some of these are shown in the bottom right image. So we see things like snout or jaw abnormalities, pericardial edema, yolk sac edema, trunk or axis malformations, and also caudal fin malformations. So this results in a massive data set containing information on over 200,000 individual zebrafish and a wealth of toxicity data to study. So to give you a sense of the chemical space, we compared the physical properties of the 1,059 chemicals in our data set to those found in the Integrated Chemical Environment, or ICE database. While these chemicals are only a fraction of all the represented chemicals, they represent a broad range of physical chemical properties as shown in this principal component analysis. And so you can see further that in our training and test data sets, the training sets are shown here in the red. And while they don't represent the full chemical space, they do a good job of at least covering a good portion of it. And then we have our blue chemicals, which are our test set chemicals, that partially overlap with our training set, but also have some distinct features that we were hoping that the model could predict. So the reason this test set was chosen was because it was conducted at a temporary different stage, so years after the initial screening. And so these were kept out just to make sure that we can continue to make predictions based on new information as we continue to screen. So now leveraging the chemical information and toxicity data in our database, we can attempt to make toxicity predictions of untested chemicals. So for our analysis, we utilized a novel algorithm developed to map and vectorize 3D chemical structure that was originally created for use in material sciences. So to take advantage of this method, we retrieved 2D chemical information from the EPA's chemistry dashboard, then used OpenBabel to convert these 2D structures to 3D chemical structures and stored them in PDB files. And so this is just an example of some of the structures in our data set and a visualization of their 3D structure. So we can get sort of, these are just some of them, we're showing a lot of the benzene rings here, but there's a variety of chemical structures. So finally, as shown in this box here on the right, we took this 3D information that was stored in these PDB files and then converted it to a weighted set of points that retained all the information of the original chemical structure, but now in a matrix form. So this is an example just showing a chemical of methyl isocyanate, which is an antimicrobial pesticide. And so in the PDB files, we can see that each of the chemicals are listed. So we have our carbon, nitrogen, sulfur, and then 3 hydrogens, and then their positions, X, Y, and Z coordinates. So what the algorithm does is it takes each of the chemicals in turn and sets them as the 0 point or the center of the 3D structure. And so each chemical has a unique identifier based on the periodic table, so which period and group they're in. So this one is carbon in period 2, group 14. It's set at the center of the 3D structure. And then the next closest chemical is built into or added to the matrix. So here we have the nitrogen, which is this one, is the next closest to this carbon. And it's put onto the x-axis. And then we go onto the sulfur, which is the next closest atom, and is put into the matrix, and moving down the list. So then we do the carbon, and then finally the hydrogens. So then we just move down to the next chemical in the PDB file, which is the second carbon. So it's this one. And so now the closest atoms are hydrogen, and then followed by the nitrogen, the carbon, and finally the sulfur. So that's how we convert the 3D structure information into a 2D matrix, but retain the 3D information. The last thing we do is we have a weight matrix, which says how common a particular structure is in the molecules, how often a structure appears, and it's assigned a weight. Pointer is not working, but okay, there we go. So the structural information is then used as input to train 2 different types of neural network generators. So here I'm showing the 2 different, the 3D converted matrices, and they're fed into this deep neural network generator with 2 serial fully connected neural networks. So we have V sub I, which is our views matrix, and then we have W sub I, which is our weight matrix. And so then we ask the generator to produce a toxicity matrix of the 18 or 17 morphological endpoints, and then this little mortality endpoint as well. This generated matrix is then compared with the actual empirical toxicity matrix, and the darker shades here indicate those that have higher toxicity values. And so the goal is the network is trained to minimize the difference between these 2 matrices. In a similar manner, we utilized a conditional generative adversarial network architecture, which takes chemical structure information and feeds it through two fully connected deep neural networks to create a predicted or generated toxicity matrix. Then the chemical features identified are fed along with the empirical and generated matrices into a second fully connected discriminator, which is there to distinguish between the empirical and the fake or generated matrices. And as the adversarial game continues, the generator gets better at distinguishing between these fake and the real. And then as the training progresses, the generator becomes better as well. So this is a way to also, once again, train a network to make accurate predictions based on structure. So training results shown in figures A and B in the top left show training plots for the deep neural network in panel A. And this is titled GoZT, or generator of zebrafish toxicity. And you can see it takes significantly less training time with only 75 epochs to really reach a good level of training loss. Whereas in panel B, titled GAN-ZT, it takes much more, almost a little over 2,000 training epochs to really train this network. So the GAN requires a lot more computational time. So following training, the models were compared with other commonly used machine learning methods. These were trained with the toxicity data, but instead of the 3D chemical information, they were trained on 2D chemical fingerprints, which are commonly used in the industry. And so these methods included support vector machines, multi-layer perceptron, random forest, and then we have the two neural networks that we've been talking about. So the results in table 1 show that the GoZT significantly outperformed all other types of machine learning architectures when we use kappa and the area under the receiver operating characteristic curve as performance metrics. So while on the other side, the GAN significantly underperformed the others. So we next investigated whether an ensemble approach combining two of these models in table 1 resulted in improved performance. So what we did is we combined the best model, which was the GoZT, and then each of the others in turn. So GoZT and the SVM, GoZT and the MLP, etc. And then we looked at positive predictive value, kappa, and OROC as the primary measures of performance. And so one other thing I'll note is that this is performance based on evaluation of the holdout or test set chemicals following training on the training set. So by combining the models, only the GoZT, GAN-ZT combination resulted in improved predictive results across all three metrics, with significant improvement in PPV, kappa, and small improvement in OROC. So you can see that individually, we get decent performance for GoZT, but in combination, we really do see a marked increase in predictive performance. So as shown in the Venn diagram here on the right, just to orientate you, we have in our test data set, there are the 59 chemicals, and of those 7 are actually active, so actually have a toxic effect. And now we've asked which of the models does the best job of predicting. So you can see that both models, there's 2 active chemicals that it misses. It captures 5 of the 7, but then we have a lot of false positives, or a couple of false positives. And so the combined model in the GoZT removes 4, and the GAN-ZT, we have the combined model removing nearly 20 false positives. So in conclusion, our results show that deep neural network-based generators are capable of predicting zebrafish toxicity using a novel 3D chemical structure algorithm. Our deep neural network generators greatly outperform other machine learning methods, as well as our GAN-based generator. But a combined GAN and deep neural network ensemble model results in a large improvement in positive predictive value and a small increase in kappa and OROC, resulting in a good predictor of empirical zebrafish toxicity. So now that we have our trained model, the next steps includes creating a priority list of chemicals for in vivo testing and empirically validating these results. So another avenue that we're excited to investigate as part of this is the development of a deep learning methodology to evaluate chemical mixtures. So with that, I would like to acknowledge my postdoc mentor, David Reif, for supporting this project, as well as our fantastic collaborators at Oregon State University, Robin Tangway and Lisa Trong. I'd also like to thank members of the Reif Lab for all the insights, and the NIEHS and NCI for funding this research. So thank you for your attention, and I'm happy to take any questions.
Well, thank you so much, Adrian. Let's start with the question and answer session. So judges, Juba, yeah, please go ahead. You have a question?
Yeah, I have a question. Nice talk, thank you. My question is that why in the beginning you decided to use 3D chemicals structure descriptors instead of 2D? Because usually QSAR, we found that for 2D seems to perform better than 3D in most QSAR models. Thank you.
Yeah, so it's a good question. And so, industry standard is using 2D chemical structures, and that's what QSARs and read across and everything, a lot of it is based on. But we don't live in a 2D world. So that's kind of why we sort of focused on 3D chemicals to actually be able to, based on their structure, could we predict toxicity more accurately? And that's also why we compared our 3D model to existing 2D models. What we haven't done is actually compared using a deep neural network with 2D based modeling versus 3D based modeling. So that is a really good point and something we should follow up with. But based on the initial findings of how with a multi-layer perceptron, which is a type of deep neural network trained with 2D chemical structure, we didn't see as much improvement, or it didn't perform better than our expectations for the deep neural network with 3D chemical structure. So hopefully that answers the question.
Thank you.
Oh, thanks, Adrian. For the audience, please keep the question coming at the Q&A box. And Catherine, you have a question as well?
Yes, so really enjoyed it. That's fascinating. So in the zebrafish model, is it very standardized as to stage of development and the experimental protocols, so that it's probably a silly question, but basically it's predicted exactly the same each time?
Yeah, so the methodology for when we expose and when we actually assess is pretty standard across zebrafish, labs and that kind of thing. So normally between 4 and 6 hours is when you start the exposure, and then you'll commonly do an assessment at 24 hours to see gross mortality or any major effects. And then as they go out to 5 days, you see more of these subtle effects cropping up. So that's sort of like the standard approach, is to assess those 2 different points. And so that's sort of the approach that we've used, and that has been used for high throughput. The other thing with this is that it's manually scored at this point. We don't have any sort of computational systems or cameras that are actually doing it. So it's some person sitting there and scoring all of these manually.
And is it single sex, or are there separate experimentations for male, female?
Yeah, right now one of the limitations is at this stage of development, we can't tell sex differences between the larvae. They're too small. So we have a mixed population.
Okay. And Max, can I ask one more? Again, okay. So this might show my ignorance, but in some projects, tell me about, as you have the training and learning in the various epochs, do you have any perturbations you introduce for relearning? Like, do you ever have plateaus in learning and then you have to push the system a bit?
Generally, that's done. You do see that, and that's what you'll do, something called optimization. And so, and hyperparameterization. And so I didn't show all of the details of which hyperparameters we chose and all that kind of stuff, just based on limitations on time. But we did run through we took random subsets and ran through a bunch of different hyperparameters to find the one that was really going to give the optimization. And so we don't really see like plateauing and then suddenly another drop when we're talking straight like regression type training is the deep neural network generator is using. Whereas that's definitely an issue with the GAN, is that we see this sort of fluctuation of it hits a plateau and then suddenly it'll go down, and it'll plateau, or it'll spike up again. So that's something, because as the 2 networks are sort of trying to find their sweet spot, you might find that you're sort of falling into a local minima and you haven't really reached a global minima yet. So you have to jump to that new spot where you can actually sort of find that sweet spot again. So yeah, that's definitely a particular problem with training GANs, more so than the regression generators.
I had one more, but we can let somebody else go.
Yeah. Junha, you want to go ahead? Yeah.
Oh, either way, thank you. Yeah, I'm just curious how you're going to evaluate the prediction for a series. I remember for Tangre's data set, there are about 17 malformation sites. Is that right?
Yes.
And I think some of the malformation sites are pretty, very few responses. Yeah, and how are you going to evaluate that compared to the one that is have a lot of hits? Yeah.
Yeah, so that definitely is a challenge, and it's a good question. So the issue is that you can have a particular compound that may not have a lot of morphological impacts, that might impact one particular subset. Or the other option, or the alternative, is that when you have the large data set, we have like 1,059 chemicals, and only 166 of those are actually active. And so part of the issue is not sort of having issue with like mode collapse, where it's just going to predict everything as non-toxic. And so these are some of the things we've tried to instill into the model to try and account for that. And as far as like the training, that's why we've tried to find things that we can regress against that are really robust. And so if we have a model system that has only one morphological endpoint, the model does a pretty good job of capturing that particular one and flagging it as active. And so the other thing that I didn't get into the weeds of is, but what we're using to actually classify a group of chemicals or a particular chemical as active is using an aggregate entropy score. So we're taking all of the zeros and ones across those different 17 endpoints, and then using a Shannon's entropy-based aggregate entropy to calculate how robust the response is, and then we predict that aggregate entropy as our predictive value of activity. So thresholding is also another that's part of the technique we've used to adapt to that.
Okay, we have 1 minute left, so I guess we have time for one quick question. Yeah, anybody?
If no one else has anything, I'm going to want to revisit a question that was asked earlier, and I'm just going to ask it in a way that I hope I can understand. Is that the thrust of your talk is that 3D modeling is better than 2D, but I took that previous questioner to imply that other people had seen that 2D outperforms 3D, and I'm just trying to unpack that. Is a fair restatement of your answer that the model is part of the reason that the different inputs were behaving differently? Just help me understand that.
Yeah, I think that there's two reasons why the 2D information is used. It's one, it's easier to use, so you have less information, and two, I think traditionally they've found good robust results from it, and so that's why sort of it's been run with. It performs, almost as good as the 3D, and it's just much easier to do. And so that's where why 2D has been utilized. And so in our case, we tried the 2D with a multi-layer perceptron, which also is a deep neural network, and we compared that to our deep neural network with 3D. And while the information is a little different, the underlying hypothesis or methodology architecture is very similar. And so I think that the takeaway for me is that the 3D will probably outperform the 2D because there's more information and we can capture more detail about how the compound space is working. And so that's where like having a robust information on 3D positioning really comes in, and as more NMR data becomes available and we have more accurate structure information, I think that'll become more prevalent.
Right. Well, thank you so much. So with that, we'll end the Q&A section. Thanks again, Adrian, for your presentation. Let's move on to our first speaker. Jay West from Cytovision will be talking about his work on transcriptomic meta-analysis reveal a molecular fingerprint underlying hepatic cholestasis response. So thank you, Jay.
Thank you, Max. Let me make sure that you all can see my screen. How does this look on your end?
Perfect. Thank you so much.
Okay, great. So hi, everyone. I'd like to first say thank you to the organizers, Max, and everyone at EMGS for putting together this excellent conference and competition. I've been really blown away by the science so far today. This has been really great. My name is Jake Reske. I am a graduate intern in toxicogenomics at Cytovision, LLC. which is a small chemical safety organization based out of Research Triangle Park in North Carolina. And I'm also a PhD candidate at Michigan State University in the Genetics and Genome Sciences program. So today I'm excited to share with you the work I did with Cytovation as an intern on developing a molecular fingerprint for predicting drug-induced cholestasis. I think this talk is going to be a little bit more biological, but definitely with some modeling built in as well. First, I wanted to give a platform for what cholestasis is and why it's important. This is a complex hepatic bile acid metabolic disease that results from altered metabolism that leads to systemic accumulation of bile salts. This disease can either present as an acute condition or chronically, depending on the mechanisms. And clinically, patients present with symptoms such as pruritus or itching, jaundice, abdominal pain, dark urine or pale stool, and fatigue. And if left untreated, cholestasis can lead to death. And clinically, there's a hallmark diagnosis of elevated serum bile salt levels as a hallmark of this disease. As I alluded, this is quite a complex and multifactorial disease that can ultimately lead to carcinogenesis if left untreated. There are a number of characterized environmental, genetic, and cellular cues to cholestasis that can lead to its pathogenesis. And the focus of today's talk will be drug-induced cholestasis, of which a well-fleshed-out mechanism is bile acid transporter inhibition through xenotoxicity. As a result of cholestasis, there are a number of pathophysiological, cellular, and molecular kind of results and cascades that can stem from bile acid toxicity, of which the parenchymal cholangiocytes, as well as hepatocytes, immune and stromal cells can all contribute toward its pathogenesis. And again, ultimately, if left untreated, this can contribute to cancer development, such as hepatocellular carcinoma or cholangiocarcinoma. The focus of my talk today is drug-induced cholestasis, which is a really important public health concern, as well as in the drug development space, because this is a common reason for compounds to terminate during clinical trials or even after they've been approved for use on the market. So here's a list of various hallmark papers dating back to the 1950s, in which compounds that were either in phase 2 or phase 3 clinical trials or had already been approved for use on market had examples come up in cases of sometimes severe or even life-threatening cholestasis that ultimately led to failure of that compound in the market. And it's estimated that between 20 and 30% of drugs that fail clinical trial development or are removed from market after approval are due to hepatotoxicity. So this is, again, a widespread issue in chemical safety as well as in the commercial drug development space to eliminate such compounds from drug development pipelines. So overarching goal that I was lucky enough to spearhead during my time at Cytovation was to use public transcriptomic data related to experimental cholestasis models in order to establish a statistical approach that could robustly predict if a sample is cholestatic and thereby a hepatic carcinogen or not based on gene expression measurements. So in drug development or safety studies, such a model that we aim to establish could be applied to cell-based transcriptome measurements following the exposure to an unknown compound of interest with the resulting probability that that compound is cholestatic. So we aim to establish a commercial assay that could be used by the community for screening and eliminating cholestatic compounds or raising their alarm. And the data I'm going to be showing you are kind of the first phases of this project that I was, again, able to drive during my internship. So the first page of this project was compiling public transcriptomic data sets for a meta-analysis. And so I manually searched and curated primarily the literature and NCBI gene expression omnibus, or GEO, in order to identify public experimental transcriptomic data sets involving cholestasis or known cholestatic inducing agents that were available. And on the right is the table that I was able to compile of usable data sets that were publicly available that included 10 GEO accessions from 8 peer-reviewed studies. And all of these were then processed through a uniform computational pipeline that was totally wrapped in R. As you can see, these are quite diverse in their nature in that we have both RNA-seq and expression microarray data sets, mouse, rat, and human model systems, both in vivo and in vitro data, and a number of different strategies for inducing or modeling cholestasis, including drug treatments, genetic models, surgical treatments, and dietary. Here's an example or a list of those papers that these transcriptomic data sets originated from. These are from many of the leaders globally in cholestasis across the globe at academic laboratories. And we think that we did a pretty good job at identifying and leveraging the major players in the field and the major available data sets that we had on hand. So as you might imagine, the first major analytical challenge we faced in this study was to understand how we can usefully integrate these diverse biological systems, being mouse, rat, and human, and in vivo and vitro, across, again, variegated data types, microarray and RNA-seq, for example. And we actually had a very simple strategy, which was to first take all of the data sets and convert them to the same set of human gene orthologs, and then z-score all of those log gene expression measurements across the relative transcriptome. So for example, on the right, if you envision a given transcriptome sample, and we look at the expression of gene J in a given sample, maybe gene J is expressed 2 standard deviations above the mean of all genes in that sample. So we would give this a expression score Z of 2, and we would use that for our meta-analysis. So this was the strategy we had on bringing all of these heterogeneous data sets together. Here's some examples of gene expression measurements in cholestasis that we compiled in our meta-analysis. On the left is an expression microarray data set in which mouse underwent a common bile duct ligation surgery to induce cholestasis versus a control or sham surgery, where we observed upregulated expression of the CD14 gene in those cholestasis mice. On the right is an RNA-seq data set from human patient liver biopsies from patients with the cholestatic chronic syndrome, Alagille syndrome, compared to patients with control livers or non-cholestatic patient biopsies. And we see that these livers had elevated levels of the CHEK2 gene. So overall, across our meta-analysis, we observed and measured 30 experimental cholestasis versus control type internally controlled experimental comparisons that we analyzed. Our first major question from the meta-analysis was, are any genes commonly misregulated in cholestasis? So we define differentially expressed genes as having a z-scored log fold change value greater than 2. In other words, this would be genes that have a 95% expression alteration in cholestasis, 95% greater than the mean of all genes across the transcriptome. And we were somewhat unsurprised to see that we actually didn't identify any genes that were commonly up or down regulated across all cholestasis data sets. Overall, as one might expect, there was significant and substantial heterogeneity observed across our meta-analysis cohort. However, we did identify 28 genes that are upregulated in at least 8 out of 30 experimental comparisons, and 19 genes that are downregulated in at least 8 out of 30 comparisons, as shown here on these heat maps on the left. We then did some gene set enrichment analysis of these genes affected in cholestasis. Among the consistently upregulated genes we identified, there were a number of stress-related pathways and cell cycle G2M checkpoint P53 type pathways that were upregulated, signifying that these, as one might expect, these cells are experiencing stress and a lot of cellular turnover due to bile acid toxicity. Whereas the consistently downregulated genes, while there were only few that we identified, it was a nice reassurance that we saw bile acid metabolism was suggested as an enriched pathway, even though we only identified 2 genes belonging to this hallmark pathway, those being DO1 and a CYP transporter. But this suggested that we are possibly retrieving true biological signal out of this meta-analysis that's related to bile acid toxicity. The major focus of this project, though, was to see if we could use these transcriptomic relative gene expression measurements to predict if a sample is cholestatic or not for an assay. So in this machine learning type workflow, we then took roughly 200 transcriptome samples from our meta-analysis and computed the relative expression values for over 12,000 genes. And then we performed our z-scoring to standardize them. And then we used a number of feature selection approaches to narrow down our feature space to 90 genes for predictive modeling. We then used a pretty straightforward machine learning framework where we employed Monte Carlo cross-validation sampling, in which we randomly sampled 70% of the samples for training models, and then the remaining 30% for testing models. And then we iteratively sampled this many times to gauge an idea of model performance across this heterogeneous data collection. And then we used some various machine learning approaches, namely just some regression models, and finally, using cross-validation primarily, measured our sensitivity, specificity, and accuracy. Our first question was, is there any transcriptome-wide signal that may be able to distinguish cholestatic versus non-cholestatic samples? So we leveraged random forest, which essentially allowed us to throw the entire transcriptome at a model and ask if any of these gene features appear to be predictive. And it did suggest that it's likely that there is signal that can distinguish the cholestatic versus the non-cholestatic samples, even in this heterogeneous environment. Our random forest classifiers had roughly 82% cross-validation accuracy. And on the right here, you can see a number of genes that were deemed as important to the cross-validation in our random forest model. But for a commercial assay, we really were interested in defining a small gene expression panel that we could use to predict cholestasis sample as an assay again. So we employed a very straightforward logistic regression where we had samples labeled as cholestatic or non-cholestatic, and we searched our reduced feature space exhaustively with a best subset search. And we were actually really excited to see that the expression of only 6 genes could be suggested to predict cholestasis samples by cross-validation with roughly 84% accuracy. So this is substantially better than random 6-gene models or random classification. On the right here, you can see a ROP curve showing the sensitivity and specificity of our model with an AUC of above 90% for a given cross-validation seed. So we're really excited that, again, with this really heterogeneous cohort of samples, that we could predict, at least these data suggest we could predict with a model whether or not a sample is cholestatic or not with over 80% accuracy. And we finally wanted to try to push this a little bit further with more transcriptomic features to see if we could improve accuracy. So we then employed just a simple greedy search where we exhaustively then searched the transcriptome for additional genes that would improve this model performance, our logistic regression model. And we saw that our best subset 6-gene expression model could predict with roughly 84% accuracy by cross-validation. But if we added a 7th gene screened across the transcriptome, we could achieve a few % increase in our accuracy performance. And we did so again with an 8th gene model, and until we saw performance plateau around 9 genes, where we saw expression of 9 genes could predict cholestatic samples with roughly 88% accuracy based on cross-validation. And so finally, here's an example of the model performance of a cross-validation sample seed that had roughly 88% accuracy. So this is the output of our model that we're hoping we could eventually see on real-world data, where cholestatic samples had probabilities mostly above 80% as being likely cholestatic, and our non-cholestatic samples had mostly below 50% probabilities. But as you all know, I'm sure this is simply just a cross-validation type of approach. We really need to test this on an entirely new set of samples and probably refine and validate this model. So our future efforts are now to generate in-house drug exposure data in a human hepatocyte model that we can refine and possibly rebuild this model in a more controlled system for a commercial assay. So I hope I've convinced you all that there's a substantial need to determine if an unknown compound is likely to induce hepatic cholestasis in the chemical safety and drug development spaces, because many compounds are pulled from the pipeline or market after sometimes rare cholestatic indications. We've shown that meta-analysis can be biologically informative for defining cholestasis gene regulatory networks and predictive modeling, even across diverse experimental models, organisms, and assay platforms. And finally, cholestatic gene expression signatures, we believe, can be predicted through expression of only 9 genes with an estimated 88% accuracy. But our current work is seeking to validate and improve this. So we're hoping to eventually someday have a commercial assay come from these work. And I really appreciate you all taking the time to listen to this work today, and I'm happy to take any questions.
Right. Thank you so much, Jay, for the presentation. So let's get started with our question and answer section. Kathleen, you have a question? Oh, you're muted.
You think I'd have that figured out by now. Okay, that was really super is all I was saying. So that, now I was wondering, can we fool it with some particular phenotype that's very close to this? And with data, see just how well it does in distinguishing phenotype?
Yeah, I think this is a really excellent question. So I didn't really have time to get to this, but there are actually a number of hepatotoxic phenotypes that are non-cholestatic. So this was a major consideration we had kind of moving into this was, can we distinguish hepatotoxicity that's cholestatic and hepatotoxicity that's non-cholestatic? And we do have a number of samples within this meta-analysis cohort that, as I'm sure you can see, is quite rough. But we do have some samples in there that would fall under this category of being hepatotoxic, but non-cholestatic. But really, in our future efforts to generate our own data and refine this model, we are including a number of samples, about a quarter of our drug cohort, that will make up that, that category of being hepatotoxic, but not within this cholestatic type phenotype. And so we're hoping that we can kind of tease apart that signal a bit. That's a great point.
So they'd go into your training so that, okay, that's really super. Do I get to ask one more?
Oh, sure, follow up.
Yeah, so when you're adding your genes back in, is it more genes is better, or in testing particular genes when you go to 7, 8, and 9, it actually matters which gene you're putting back in?
Yeah, absolutely. So what I did was we had, 6 genes that I'm not going to reveal, but I will say they have, some of these have known links in liver biology and cholestasis. So that was nice to see. But, we have our 6 genes that we've identified. These are predictive. We want to see if we can improve this. And so, yeah, we screen all 11,000, 12,000 other genes, and we see that a lot of those don't do anything to model performance. But there are some genes that do add a substantial amount to performance. So kind of my strategy was, let's take our 6 gene model, screen all of them, and then take the best one and bring it in. Now let's repeat that again. We have our best 7 gene model, screen, find the best predictor, add it into the model, and so on and so forth. And in the data I presented today, we saw a plateauing at around 9 genes, suggesting that we didn't identify any further genes that would add substantial information to the model.
And I had one more. So in your ratio of your trained test of your 70-30, if we go back as you're sort of integrating your transcriptomic data across different model systems, are they mixed across your training and testing so that you're covering all of them, or did they happen to be within the 70-30 split?
Yeah, so again, the data I'm presenting are pretty preliminary in this study. And simply what we had was we had these 200 heterogeneous samples, and any one sample of that is probably going to be pretty heterogeneous. So the way we did it was we did a lot of 70-30 splits, and then we averaged over all of those. So we did, in a lot of these performance studies, about 1,000 random samples. And what you're seeing is the average of all of those.
Mm-hmm.
But yeah, ultimately, to get true model validation, we need to have a separate set that we're going to use for kind of final positive predictive value. And kind of in our further iterations of the study, as we're generating our in-house data, we're going to be reserving a sector of those samples that are representative of the space that are only used for model validation, that would not be used in any of the training purposes. Yes.
Thank you.
So we have 4 minutes left for the member of the audience. Please feel free to submit your question at the Q&A box. And Tom, you want to ask the next question? Oh, the mute.
I believe you're muted.
I am indeed. All right, let's start again. So I'm trying to unpack something I maybe didn't get, is that the data sets that you went and mined, these are biosamples from like livers and actual, but the goal is to apply it to an in vitro toxicology paradigm. Is that what I should be understanding, or help me out if I'm not getting it?
Yeah, so it is. And I would say the rationale behind that is resources. I was an intern at a company, and we wanted to see what we could kind of spearhead on such a project and see if we could kind of extract any gene features that might be relevant to this project. So yeah, the ultimate end goal that we're now seeking to get into is, yes, an in vitro cell-based assay where a customer would come in, have their compound, we'd have our cell-based model, they'd screen it, and we'd have the predictive value. But at this point in the study, we were ultimately trying to see, can we leverage any existing data already to produce some sort of a model that can extract and differentiate signal that's cholestatic, maybe that we can use as features in an eventual commercial assay. But yeah, this is a highly exploratory phase of the study in which we are trying to understand what biological signals may differentiate, what gene expression features can work, and is this possible at all? Can gene expression differentiate cholestatic samples?
Yeah, thank you, Jay. I believe that we have one question from the audience. So the question from the audience is, Jay, I don't know if you can see it on the question and answer box.
Yes,
but,
I can.
yeah, okay. So he's asking about was Z-score or log2 fold change used as input to random forest? Yeah, would you mind responding to that?
Yeah, absolutely. So for the random forest, when we had our samples, we had relative gene expression values that included both RNA-seq and microarray, and from different platforms. And so our strategy there was essentially we do some normalized count transformation, and then we z-scored those. And so we would have a sample-to-sample z-scored measurement of relative gene expression transcriptome-wide. And again, that was kind of our naive strategy in compiling mouse, rat, and human, and in vitro and in vivo data sets. We are aware that there are probably some biases that as a result of, but again, this was us trying to leverage just available data that's out there, and this was one strategy we had.
All right, thank you, Jay. I believe we have one comment in the Q&A, and I guess I'm going to ask Stephen's question. So beside an in vitro model, are you considering a rodent model as well?
Yeah, so a major focus of Cytovation's kind of motto or goal is new approach methodologies that avoid in vivo studies. So a flagship of kind of their products are in vitro based cell-based models. So we are trying to, at least for this assay, and for a number of different reasons in terms of cost as well, trying to develop something that would be truly cell-based. However, perhaps in later stages of this and validating such approaches, maybe in vivo rodent models could be used somewhere along the pipeline, or as we think about extending this framework to other applications, maybe other endpoints that can't be modeled in vitro. I think that a similar framework could be applied to rodent models, yes.
Okay. So I believe the Q&A session is time's up. So what I'm going to do is I'm going to copy and paste the, actually we have 3 questions right now on the Q&A box. I'm going to copy and paste all of them in the chat box, and we would proceed with the 4th speaker. So thanks a lot for the presentation, Jay. I appreciate your time. Yeah, thank you so much. Okay, so our last speaker for today is Kai Wong from the University of Michigan, and he's going to be speaking to us about his work on exposome target prediction with coupled matrix-matrix completion. Thank you so much, Kai.
Okay, thank you. Can you see the screen, right?
Yes. It's me switching to. Yep.
Okay. Yeah.
Thank you so much.
Please. Okay. Please remind me if you. Yeah. Please remind me if you cannot hear me clearly, because my internet looks like it's not very stable this afternoon.
Right.
Okay. Yeah. Thank you for giving me this opportunity to show this ongoing project. So my talk is titled as Exposure Target Prediction with Coupled Matrix-Matrix Completion. And the exposure is kind of defined as measurement of all exposures of individuals in a lifetime and how these exposures relate to health. And measuring the exposures and investigating the corresponding linkage to health effects is a challenging task. And especially given there are like 2,000 new environmental chemicals being produced each year. As shown in this figure, so those chemical environmental exposures will like kind of affect the internal environment on our DNA, protein, or or other stuff, and then further affects the human health. But a good thing is there's a lot of exposure-related biological databases were established. For example, for the CTD and the LINKS program and the chemical dashboard from EPA and the T3 database. So from this talk, so all the data is from the CTD database. And a key point to investigate the health effects from exposure is to understand the molecular targets of those exposures. So the logic here is like how to predict the environmental exposures, the targets of those environmental exposures. So this topic is basically focused on this prediction. And for the target tissues, this is in our future plans, and also the health effect in our future plans. And so far as we know, so there's no method that's been developed using the current knowledge of chemical target interactions and their relationship among the chemicals and the gene targets to predict novel exposure target interactions. But similar method was developed used in drug target interaction predictions. And so within these methods, so previously method was developed for drug target interaction predictions, like includes like a matrix completion algorithm and deep learning algorithm and some other like distance-based method. So for drug target prediction, drug target interaction predictions, because the drugs are usually specifically defined for a specific target. So those methods are usually use a binary value, like a 1 or 0, to indicate the interactions between the drug and the targets. But for the environmental chemical exposures, so since they are not designed for target genes and stuff, so we think that the use of binary value is not a good way to represent interactions. And so that's why we want to use this coupled matrix completion method on the target environmental chemical and target predictions. And within those methods, so the matrix completion method shows like a very good performance on drug targeted action predictions. So the matrix completion is a task to finding the missing values from a partially observed matrix, which is something like this. So if we have a matrix, we have some missing values. So those missing values can be imputed based on the existing values in this matrix. So within this kind of method, so there's one group of the method called the matrix factorization method, which is very popular and shows great performance on this. And so this method basically is decompose the matrix like this matrix A here into two small matrices, and then the missing value can be imputed based on the multiplication of the Y matrix and Z matrix here. So the limitation of this of those group of method is that they are kind of only focusing on the existing values in the A matrix here. So in this method, so actually it is a new algorithm that was developed in from our university. And so this method is basically we are not only using the existing data in the A matrix here, but we also included the two coupled matrices. So one is coupled to the row, one is coupled to the column. So in here, the A matrix, it's a partial observed matrix, so which is like M chemicals by N genes, and the R chemical here, so R represents row. So this row coupled matrix is a symmetric matrix which contains the chemical similarities, and the C matrix here which contains the gene similarities. So predicting the missing value in A is the final goal. So this algorithm, so this is the input for this algorithm, and output will be like 3 matrices with exactly the same size to the input. And we call it like a B1, B2, and a sigma. So the algorithm have like 3 steps. So the first step is to pre-process the coupled 2 matrices, which is like we have 4 different hyperparameters here to pre-process the coupled matrices. And for the second step, so second step is a for loop, as a main loop. So after we finish the pre-process step, so those 2 matrices here, and so here is a toy example of the matrix A. So those 3 matrices will be used in the iterations to the calculation. So we will initialize the B1 and the B2 as 2 identity matrices and grab all the existing values from the matrix A to create this vector and these 2 column matrices. And those 2 things will be used to generate like it's kind of like constraints in the calculation, in the iteration. So from here we can see, so those two coupled matrix, pre-processed coupled matrices will be directly used in the calculation to find the values for updates the values in B1 and B2. And those two things will be, it's kind of the constraint to make sure that algorithm will grab the corresponding value from the coupled matrices in the calculation. And after the calculation, so the sigma will be updated by B1 and B2. And then when we have all those three matrices generated and multiply them together, we can have the matrix A, which have all the predicting value in there. And compared to the original one, we can test the performance. And yeah, so for now, all the data I used is from the CTD database. And so this is total number of the CTD, total number of chemicals and full number of target genes and the interactions from the CTD database. So we can see, if we put those target and chemicals into a matrix, so the matrix size is very big, but the known interaction values only took like a 0.2% of the total matrix, which makes that this matrix is like a very sparse matrix. So for the following result, I'm basically focused on human data. So for the human data from CTD, also we can see the total size is kind of big, but the known interaction is only took like 0.2% of the total size, which means that also this matrix is very sparse matrix. And also in CTD, there are some drugs included. So we filled out the drugs, and here's the number of the, after filling the drugs, here's the number of chemicals in this database. And so for here, so because our algorithm need like a 3 matrix to do the imputation. So for here, first we need to define the coupled matrix. So for this matrix, is we use the, so this is like the gene similarity. So we use this this R bioconductor package to calculate the gene similarity based on 3 different Go terms. Yeah, and for the chemical similarity matrix, we use RDK to calculate the Morgan fingerprints and the topology Torsen fingerprints. All those values are located within 0 to 1. And then to define the main matrix, which is A here, so actually we calculate the numbers into 4 different ways. So for N1, it's like we just count how many chemical and gene pairs are there, and normalize them into a range like between 0 and 1. And N2 is like the normalization log transformation based on N1. N3 is the count of the PubMed ID of those chemical gene pairs. And N4 is a log transformation based on N3. And yeah, so this is for the value in the matrix A. And for testing and comparing to other methods, we manually define a golden standard data set. So for the positive data set, it's like we select some gene chemical pairs that N1 is larger than 0.85. And for the negative golden standard data set, we select some of them, like 20 most studied chemical without any evidence of gene target genes. And when we actually generate the data to test the algorithm, I generate 3 sizes of the matrices, which is 200 by 400, 400 by 6, and 6 by 8. So here's the chemical by genes. And actually to do the testing, what we did is here. So because we have the golden standard data set here, so basically we will randomly select a small portion of the interactions, the chemical target pairs from the golden standard data set. If it's a positive data set, we will replace those values into 0. If it's negative, so it's 0, so it remains 0. Because we have like 2 different kind of similarity for the chemicals and 3 different kind of similarities for the genes. So in total, we will have like a different 6 different rounds for the calculation. And here is the result. So basically, because we have the golden standard, so we calculate the ROCs, AUCs on the testing data set. So here is 200 by 400, and so it's N1, N2, N3, N4. So from here, we can see like the different normalization method shows a different performance. It's like N2 have the best performance. And also from here, we can see the converging is very fast. Usually it's like a 2 iteration is sufficient from here. And after that, so here is we also compared the input size. So how the input size like affects the result on the EUCs. So because N2 it has the best performance. So for here we use N2 to calculate on the 200 by 400, 400 by 600, 600 by 800. And initial finding is like increasing the size of input matrices can slightly improve the performance, but it's not that exactly. So compared to other method, so we compare to CMF and WNN because it's an ongoing project. So I'm still like comparing to other methods, but those two methods is kind of very similar. But those two methods is also like a design for drug targeting action. And for here, so this is the result. So we apply our method and those two methods on the 200 by 400 and two matrices. For our method, there's two iterations. So we can see the AUC we have is like better than those two methods. And also the running time. So our method is comparable to the WN, but CMF is kind of slow. So after this, there's a case study. So for case study, we choose the BPA. So BPA is an important precursor for plastics, and so the endocrine disruptor and related to breast cancer is already banned and switched to BPF and BPS. And those chemicals, they have like a very similar structures. So this case study is more like a real scenario, which is like if we have a new chemical and we don't have any knowledge on this chemical, so which gene they can target. So in here, how can we like predict the potential gene targets for this chemical? So for here, we generate a matrix like a 500 by 1000 genes and corresponding couple of matrices. And for the BPA, we keep those values in here. Those data also from CTD. But for the BPF and BPS, we remove the entire row for BPS and BPF to like try to repredict those values for those two chemicals. And here is the result. Overall, so we calculate because it's like three vectors there. So overall, we calculate the Randall Kendall rank correlation coefficients. So basically, so this is like to show although the exactly interaction value have some like a distance, but the pattern, they kind of like correlate with each other, which means it's like if a high value for BPA in this for the specific gene, there's also a high value for BPS and BPF for that specific gene. And after that, so here is like the specific genes I selected. So we can see for BPA, because it's like a very well studied chemicals, and the targeted genes for E kind of like the ESRA1 and ACE genes. In our input data, so those two genes is like have a value 1, and for the ESR2 is like 0.5 or something. So from our result, we can see although the number is not exactly same, but those numbers are also the biggest number in that vector. And we can see so for ESR1 and ACE genes, they also have some values here, and the ERS2 is also like a half of the interaction. From our results. And an interesting thing is that for BPA, there's NOG gene is not show any interaction from CTD, but we have like found this gene might be a new potential target for BPS and BPF. And so the summary is like, so this method already being successfully applied on drug target interaction prediction with random input, but we modify the algorithm a little bit and apply this on the environmental exposure target predictions. And we believe this method can be used to predict, to perform these predictions, because we modify that so it's not restricted to binary inputs, and it's fast and optimized for sparse matrices. And in future, we can extend it to the coupled matrix to coupled tensor, which means like we can use all of those data together to predict to find the missing values. And also we believe that maybe we can use this method to predict the target tissues. Okay, that's our acknowledgement. So I want to thank my advisor, Maureen Sauter, and Dr. Keevan, Justin, and those two grad students, and also the CISCAR and our department and the Emily Center and the School of Public Health, and also my cat. He's cute, but sometimes it's annoying while I'm working from home.
Well, thank you so much, Kai. All right, so so this would be our final Q&A section. So please do, for the member of the audience, please do submit your question to the Q&A box. So so the judges, please feel free to ask any question.
Oh, I have a question, Max.
Oh, great, go ahead, Tess.
That was a really great talk. Thank you. It's really interesting work. I kind of have a big picture question or application question. So I know you started off by talking about exposomics, and so I'm just wondering if a goal or a use of this tool could be kind of used in reverse. Like, let's say you have, you use these data sets that you've been training your models on, and we know that there's gene biochemical interactions. Could epidemiologists have, like, say they have like a transcriptomic data set from a population, could you use that to kind of predict what chemicals this, like, a population might be being exposed to? So kind of like the reverse, like moving backwards in a way.
Yeah. Yes. Actually, that's the next step we will work on. And basically, because for now we just collect the data from the CTD database. So the database already helped me to like normalize and finding those values. But if we have like expression change levels, it also can be used here. So basically, just we can normalize that data, like make sure they are located in a range, and just make sure that that number is actually makes sense to represent those interactions between the chemical and the gene. So then we can use this because in that way we just replace a number in the matrix A. So yeah, we only replace this values in this matrix. And other two matrices actually is just a similarity. So yes, definitely yes, we can use expression levels.
All right, well, thank you, Kai, for the response. And for the member of the audience, please feel free to ask any question. Tess, do you have any follow-up question, by the way?
Nope.
All right, yeah, okay. And Tom and Junma, do you guys have any question? Tom? Yeah.
Hang on. My biggest question is why Tess her name appears to me to be Max. So I'm not sure what the reason for that is. But let me ask a question. I want to go back to really one of the first things you said, and you brought it back at the end. And I just want to unpack this. It's not really a bioinformatics question, maybe so much, but you've said that drug targets are modeled using binary values, yes, no. And you've argued, or at least you suggested, that you thought that wasn't such a good way to look at environmental exposures. And I'm wondering if you can rationalize the reason for that, because if they're chemicals interacting with molecular targets, why are the rules different?
Oh, so the rationale here is like, so the drug is actually designed for a specific target, right? So, but for environmental chemicals, those are not actually designed like it goes into our bodies. So that's why we think, so if we use like a binary to indicate that this chemical can target to the environmental chemicals can target to these genes, so it might be it's not a good way to represent in that way.
Is it an argument of potency that the drugs are designed to be especially potent for specific targets, or what? I'm still, if you still have a list of targets and you have a chemical and you say a drug can interact with certain ones, and are the rules different, or is it simply that you're looking for lower level affinities for targets?
Yes, kind of like find the lower affinity.
Right. Yeah. Thank you, Kai. So if any for the member of the audience, please keep the question coming at the Q&A. Junma, you have any question also? I don't know if Kevin's audio is back. So, but yeah. And Tom, if you want to ask a follow-up question, feel free to. We have another 3.5 minutes in the Q&A. Yeah. Oh,
Okay,
Go ahead.
I'll ask another question that's really, I need you to explain it a little bit more. So a lot of information came at me that I didn't really, hadn't seen some of that before. So I'm just trying to understand better in the R and C matrices that you're generating. And again, I apologize, this might be really naive questions, but when you're looking at the relationships between the genes or between the compounds, how are you deriving those relationships? I know you said this, but it went by fast.
Oh yeah. Let me jump to that slide. Okay. You mean something here?
You tell me. So
Yeah. So basically, so for here is a little bit complicated. So that's why I did a smaller amount of time on here. So basically, it's like we will use, because the pre-processed two coupled matrices will be directly used into finding to update the B matrix here. And those two matrices here is actually from those index. So the index is the position of those numbers in the main matrix. And in general, in the algorithm, in the code, is like we will generate another matrix to hold those positions. To actually in the calculation, so the numbers in those two matrices will actually updated based on that position matrix to make sure. Okay, so when I add those numbers together, okay, so the algorithm will know. Okay, so if I process the rows, so okay, so for this number, like 0.3 is here, 0.2 is here. Okay, so I will use those two numbers in here to add them into this matrix, and also we'll grab the corresponding similarities for this row and this row into the matrix.
thank you.
Yes. So basically, the position matrix is like a trick here. So let's make sure that all the corresponding values can be used into the calculation, but that is a little kind of complicated. So I didn't list a figure here to show that.
Okay. So we have 1 minute left for the Q&A. So I guess we have time for one last, quick question. Yeah. Feel free to type any question, at the Q&A box. Otherwise, Tom, maybe you can take the last one.
I'm done. Max, you haven't asked any questions yet.
Oh, I'm not going to exercise my power as the organizer. So but go ahead. Yeah. Okay. Is there any question from the judging panel or from the audience? Yeah, if not, then I guess we will conclude this Q&A and also this final round competition. Thank you so much for all our speakers and all our judges for your participation, and also thank you for spending this afternoon and evening with us. It's really great, for us to get together, in this event. Stay well. And we look forward to seeing you all in next year's EMGS Bioinformatics Challenge. Thank you and good night.